Welcome to the Canadian Herpetological Society December Virtual Conference. My name is Pamela Rutherford and I'm Chair of the Meetings and Workshop Committee. Our sessions today will be moderated by Hannah McCurdy-Adams.
Hello, everyone. I'm actually going to introduce myself a little bit first, but I'm Hannah. I'm the Reptile and Amphibian Program Development Coordinator from Wildlife Preservation Canada, and I'm currently beaming in from Vaughan, and I'd like to acknowledge that it is also the territories and treaty lands of many nations, including the Mississaugas of the New Credit, the Anishinaabe, the Huron-Wendat, the Haudenosaunee, and the Métis Nations. And so our first speaker will be Donnell to talk about a very worrying acute mortality event of painted turtles.
Good afternoon, everyone. My name is Donnell Gasbarrini, and I'm here today to share with you the story of an acute mortality event of both painted and blanding's turtles that occurred this past summer in southern Ontario. And I will be sharing this story on behalf of my entire team, who you can see here, as well as the affiliated organizations. And some of you might be wondering if this is the same old song and dance that you've seen before. And while I have spoken quite a lot about another mass mortality event, this one is a similar event that has taken place on a very different landscape. And with that, I'm going to start with setting the scene. So this mortality event took place in a restored wetland habitat in southern Ontario, a very urban area. It is an area that is highly accessible to the public and that often is visited by people who are running or walking their dogs or out with friends and family. And it saw high visitation throughout the summer of 2020 as well, despite COVID restrictions and limitations. It is an area that is home to snapping turtles, painted turtles, blanding's turtles, and northern map turtles, and where there has been quite a lot of monitoring and surveys done over the past about 20 to 25 years, which resulted in the creation of a long-term blanding's turtle Head Start project, which celebrated its 7th year of Head Start releases this past summer. And because of all of this monitoring that's been going on for some time, we do have historic data. And here I've shown the last few years worth of mortality data. And you can see that most of what we've recorded have been blanding's turtles, with some painted turtles as well. And now, some mortality, especially with regards to the blanding's turtle program, is expected because the turtles that we are tracking are juveniles, and so we know that juveniles do experience higher mortality rates than adults. However, what we saw this past summer was extremely jarring and a complete surprise. During the month of June, we found over 50 dead painted turtles. And many of you will likely immediately think, hey, that's nesting season. And you're exactly right. This mortality event took place, or it began during nesting season, and it affected mostly female painted turtles to make the situation worse. And what we thought, that we realized now was a little naive, was that once the nesting season was over and the turtles had resumed their location in the water instead of being in a more vulnerable locations on land, we thought that the mortalities would end and that we would stop finding turtle carcasses. However, you can see here that that's not what we found. Come the end of nesting season, we found a very abrupt transition in prey from painted turtles to blanding's turtles. And I'm going to zoom in on this part of the graph. I'm going to elongate the x-axis, so it's going to look a little different, but it will give us a more fine-tuned perspective on what occurred this past summer. So you can see here a very high number of painted turtle mortalities, primarily during the nesting season. So the first carcasses were found on June the 8th, and the majority of the painted turtles were found between that date and June 30th. And then on July 13th, we found over 30 dead blanding's turtles in one day. And so this was very, very startling. Here's a photo of the carcasses that were found, both painted and blanding's turtles. And the lower photo shows all of those blanding's turtles that were found on the same day, July the 13th. And so when thinking about what causes of death could have been, we sent off a subset of these carcasses to the Canadian Wildlife Health Cooperative to have a full necropsy completed on each of them, as well as some disease testing done. Now, the necropsies pointed to predation as the most likely culprit, and the testing for disease came back with no positive results. So at this point, it seems unlikely that disease was the reason for these deaths. Now, thinking about predation, we do know that there are many subsidized predators, particularly mammalian predators, that are present at this site. This includes animals like raccoons and mink, which were captured on wildlife cameras at the site, but also includes other animals like foxes, skunks, and that type of thing. And so moving forward, we are going to be pursuing some next steps. And those include gaining greater insights into potential predators at this site, as well as the dynamics of those predators. We will be using a new release site for our head-started turtles, so that we're hopefully releasing them in an area that will pose less immediate risk to them. We will be increasing the human presence at the mortality site, particularly during the nesting season of 2021. And we will be using deterrents such as wolf urine at the site, again with a focused effort around the nesting season in 2021. And this is in an effort to discourage predators from accessing the area during this site in a way that is both safe and friendly for the people and domestic animals that frequent the site. And what is the significance of all of this? Well, the frequency of mortality events is predicted to increase. Many of these events go unnoticed or are unreported. And a big motivator for me in submitting the abstract and pursuing this talk is to encourage others to share their stories. I've seen quite a lot on Facebook that these might not be as rare or that many people have experienced them before, but that they were never formally reported anywhere. And so I would just like to say, very politely, that it would be extremely helpful to get more of that information out there in a way that is accessible to other researchers and conservation staff. This will help us to become aware of the threats to species at risk, particularly those associated with mass mortality events. And it will help us to identify worthwhile conservation initiatives that can take place post-catastrophe. And with that, I would like to say a very big thank you to all of you for listening today. I would like to thank our funding sources and partner organizations and individuals. And I would like to thank CHS for hosting us all today and bringing us together in this extremely challenging time. I look forward to the questions that come forward towards the end of this session. And if anyone would like to chat further about this, I've included my email address here. So thank you all very much and enjoy the rest of the conference.
To move on to our next presenter, which is Joe, and he's going to talk to us about the effects of a frog virus.
All right, so today I'm going to talk a little bit about a project we recently finished, and it also got published. So if you're interested, Facets Journal. So basically, we looked at what the in vivo effects of a wild-type frog virus 3 and a VIF2 alpha knockout are on post-metamorphic wood frogs. And we basically looked at what are the effects on survival, body condition, and behavior. Specifically, we had 2 Rana virus isolates, which was the wild type, which was originally isolated in the 60, and then we had a knockout mutant, which basically had, so VIF2 alpha is an immune evasion gene, which was knocked out in that case. We did a BAF exposure with rather common doses. However, those doses have been shown to be lethal and cause 100% mortality in other ranids. And yeah, so we collected eggs up in the Northwest Territories, chartered a little plane, flew them down to Fort McMurray and got them shipped by Air Canada to Sudbury. And basically I raised all of the tadpoles to metamorphosis, fed them for another while, and basically we exposed them 4 to 5 weeks after metamorphosis. What did we see? In general, we did see that infection rates followed a dose-dependent pattern. However, we only saw 5% ranavirus-related mortality, which is surprising because, as I mentioned before, a lot of ranids, or in general, there's a lack of studies on adult or post-metamorphic individuals. However, normally they all die, and we only saw 5%. So something was going on there. And surprisingly, we also only saw 42% overall infection rate. And nonetheless, 82% of the individuals across the treatment showed hemorrhaging or signs of ranavirus infection. However, at the end of the experiments, 80% of those individuals didn't show any signs anymore, which is rather surprising. And on the top left, you see basically a frog which is completely clear of any signs. And on the bottom right, you see the most extreme. And basically what happens is they get those strong bleedings in the leg and in the arms. Yeah, and often you see also strong hemorrhages in the fingers. But we also assessed body condition and behavior. And interestingly, the frogs which were exposed to the knockout mutant showed a decrease in growth, but also in behavior. So that is likely caused by the fact that those knockout mutants, so that gene basically suppresses the immune system. If the virus does not have the gene to suppress the immune system, the body tries to fight the infection, right? And the immune system requires a lot of energy resources or requires a lot of power of the frog. And basically that will cause in the end that the frog grows slower, but also shows a decrease in behavior. And as you can see, we plotted that and it's quite obvious. So the dark red lines are basically the lines which refer to the knockout mutant. So what can that show to us? So basically a selection towards more resilient genetic lineages within the source population. I have to say that source population has been exposed to ranavirus repeatedly. And it could be also that it's just the immune status of the post-metamorphic individuals, because I mean, as I said, there's not a lot of studies done on adults or post-metamorphic individuals, and most of them are actually done with Xenopus. And also in general that we did not observe those high loads. It could be that those animals were infected in other organs and we only screened the liver, which is normally done, but yeah. So in summary, what does that mean? A single short-time exposure to environmentally relevant doses can cause infection in post-metamorphic individuals, but does not necessarily mean that they have to die. They can sustain sublethal infection. They're capable to clear those infections. And in the end, it is likely that post-metamorphic individuals can act as intraclass reservoir for those pathogens. I mean, we only tested it on that one population and that one species, but I guess it would be good to go on with that and expose more species to different doses of virus and see what is going on there. I have to thank a lot of people which were involved. Government organization, First Nations, because we worked on treaty territories, so Danna Nation, Smith Landing's First Nation, Salt River First Nation, and Northwest Territories First Nation, and we had a lot of people involved in that project. And that's from me, everything. And I guess I'm open to answer questions afterwards, and I hope I stayed in time.
So if you have any questions, please add them to the chat, but we'll start with a question for Donnell. How dead were your dead turtles, and do you know if it was linked to any sort of disease? And this was a question from Anne.
Thanks, Anne. The turtles were very dead, but they were freshly dead. Something that we had working in our favor is that we did have so much presence at the site, so they were being found quite fresh, and we sent off a subset of those that were found to the Canadian Wildlife Health Cooperative to test them, to do a full necropsy and test for disease. And none of the samples that they looked at came back positive with anything suspect.
Okay, and then we'll jump to Joe, and I'm sorry if you mentioned this, but how far spread is the ranavirus you're talking about in Canada, or is it here yet?
Oh, it is here. So, I mean, what we tested is the original isolate from like 60 something. But I mean, in my study area, we have at least 3 different ones based on genetics. But it is quite widespread. But as I said, like at least for me, I mean, I have rather high infection rates. However, I did not really see a lot of dead animals. I mean, it could be contributed to the fact that I work in the bush. I mean, Wood Buffalo National Park is huge. And I mean, that study, the eggs came from up north, but yeah, I mean, it is widespread, but it's not really clear how devastating it is. And if you see die-offs, it's mostly tadpoles or late-stage tadpoles. And I mean, I've seen die-offs up there which had 100% mortality, but only the late-stage tadpoles, pretty much, like never really adults. So, and that was one of the reasons why we looked into it, because I mean, I got all those positives, but yeah, they didn't show any signs. So, and I mean, what I did basically explains that yes, they can get infected at that stage, but somehow they're capable to clear it. And I mean, that has been shown in African claw frogs too. So it is possible. And yeah, I think we should not generalize across life stages with those diseases, and we should be more specific about what we're looking at. So.
All right, I've got a quick follow-up from that. Is it known whether this is a recent emerging disease, or has it been here for a long time?
Actually, there's a great paper on that we did with some other people, but we assume it came to Canada in the last 100 years. And in that area, about 60 years. And interestingly, it correlates with the build of that big oil pipeline they built up into the Northwest Territories. That's about the time frame we expected it came in, and that's just an assumption I made. But yeah, I mean, I'm gonna put the link to the paper in the chat. It's super interesting.
Sure. All right. Jump back to Donnell. Is the wetland where the turtle of mortality event occurred eutrophic? And do you see harmful algal blooms in the summer or fall?
So the wetland area, it is a restored habitat, and that work was done by Toronto Region Conservation Authority in partnership with Parks Canada. And we haven't seen anything questionable in terms of algal blooms. There is some water chemistry data being collected, though it's part of Tharusha's PhD project, so it's taking place during the winter. But anyways, we haven't noticed anything suspect in terms of the wetland or water quality.
So we'll start off with Nick, who is going to talk to us about spring peepers.
Thank you very much for having me. Seeing this time is going to be short, I'm going to just jump right in. Anyways, we all love species trees. I'm pretty sure everyone here has at some point looked at a species tree with some interest. They're really useful tools for visualizing complex evolutionary hypotheses, and they date back to the early years of speciation theory. So you can see one of Darwin's early trees from 1837 here from his notebook. But we also have a long history of using a limited number of genes, particularly mitochondrial genes, to make these trees, and we use them as proxies for species histories. But genes have their own histories of gene flow, of sorting and extinction. And we're beginning, as we start looking at larger numbers of genes and even whole genomes, to see that there are many cases where genes provide dramatically different inference from the rest of the genes that make up what we call a species. So I've been looking at the phylogeography of Pseudacris crucifer in the Lougheed lab for the last number of years. Peepers have an absolutely massive range from the boreal forests of Manitoba down to the Gulf coasts of East Texas. My predecessors in the Lougheed lab, Dr. Austin and Dr. Stewart, determined that there were 6 parapatric mitochondrial lineages that vary in age from 11 million years to 3 million years old within the species. But to date, there's been no investigation of range-wide patterns in nuclear diversity. So as part of my PhD, I set out to infill this gap. I used reduced representation genomics to get a broad sample from across the genome of the peeper and from across the range. With Amanda Cicchino, I also collected and explored patterns of advertisement call. The most notable and perplexing aspect of my entire PhD was the degree of cytonuclear discordance that I found. I found mismatches between mitochondrial and nuclear DNA in geographic extent, as well as tree branching pattern or topology. The topological discordance wasn't merely that there was a different number of lineages, which you can see here between the mitochondrial and the nuclear, there being only 3 nuclear instead of 6, but that this but this can't be explained with mutation rates alone. For example, you can see in the nuclear lineage here in the yellow that this is not discordant. The diversity all stems from a single ancestor, and the distribution and the faster sorting rates of mitochondrial DNA can explain the presence of 3 lineages. The southern lineage, however, in the blue, incorporates a crazy paraphyletic sampling of mitochondrial DNA taken from multiple ancestors. And meanwhile, the western lineage in green here stands all alone, is concordant with a single mitochondrial lineage, but not sharing its branching pattern. And so the best explanation we have for these patterns is widespread nuclear integration and replacement amongst some ancient lineages, but not others. The western lineage particularly stands out as being quite resistant to integration. And interestingly, it's also the one lineage that has a real distinction in call. So this is what we're left with, an outline. The outline here represents the presumed nuclear range, and the fill represents the mitochondrial range. There are markers for individual nuclear and mitochondrial genotypes at each point, as well as call markers of the circle ones. I think the major take home from my thesis, or at least this part of my thesis, was the need for caution when inferring from a limited number of data sources. For each of these sources of inference, they each presented an alternate hypothesis that seemed informed because that's what we were getting from that source of inference, but taken together, they present a different, novel, and even more complex story. None of them are wrong because they're all part of the species history. The early history of divergence and differentiation, perhaps in allopatry, is told by non-recombining mitochondrial DNA, and the messy history of contact and gene flow and the dissolution and maintenance of regional diversity is told by the nuclear DNA. And then the special nature of the Western lineages is supported by call data. Obviously, my sampling is incomplete. It's very hard to get everything out of an animal, but each new source of data tells a more informed estimate of the complicated history of this species. And all in all, we get closer to a whole tree, including all the braided and trimmed branches. So with that, I have a number of funding agencies and people to thank. And thank you to the organizing committee and everybody involved in all of you watching. And here's a find the peeper thing we can look at.
Oh, that's fantastic. I love these photos, and thank you for being on time. But we will move on. Oh, I guess actually, no, before we move on, I think people have had enough time. Just tell us where the peeper is. No, you have to come back later.
I'll do it in the question slide. I'll just replace me with that.
Okay. So our next presenter is going to be, well, actually the next 3 presenters are all going to talk about things on Peely Island in Ontario. And the first will be Tom talking about blue-spotted salamanders on Peely Island.
Today we're going to talk about declines in the blue-spotted salamanders on Peely Island and why we think that these declines merit a formal risk assessment and protection for these animals. Blue-spotted salamanders are a widely distributed species here in eastern North America, and at the southern range edge they actually overlap with the smallmouth salamanders. They also overlap with another unique animal called the unisexual Embystoma, an all-female lineage that possesses DNA for multiple species, including the blue-spotted salamander. They have anywhere from 2 to 5 sets of chromosomes and are obligate reproductive parasites. So they can only reproduce if they can steal sperm from one of these species that they live alongside, like the blue-spotted salamander. So here's Peely Island. This is the place that we're talking about today. Peely Island is unique because it's the only island in Lake Erie where you have blue-spotted salamanders co-occurring with smallmouth salamanders and these unisexual Embystoma. In fact, it's also the only one of these islands in Lake Erie where blue-spotted salamanders occur. The blue-spotted salamanders on Peely Island are in fact genetically distinct from the mainland Ontario populations, and this makes sense because Peely Island has been isolated from the mainland for about 4,000 years. This represents about 1,000 years of isolation. Some unpublished work by Jim Bogart has actually shown that the blue-spotted salamanders collected from Rondo, which is on the mainland Ontario, and blue-spotted salamanders collected from Peely Island actually do have a relatively large genetic distance between them. And in terms of the mitochondrial DNA, the mitochondrial haplotypes of Peely Island blue-spotted salamanders also differs by at least 5 nucleotides from populations on mainland Ontario. Samples collected 30 years ago indicated 3 places on the island where blue-spotted salamanders occurred and suggested that they made up about 6.8% of the population. 2 of those places, the blue-spotted salamanders no longer occur, and one of those places we haven't sampled in the last 30 years because it's on private land. In fact, we've only collected about 3 confirmed blue-spotted salamanders to date in all of our samples from 2015 to 2018, which is over 2,000 genotype samples. So are blue-spotted salamanders in decline? Well, it's hard to say, but we do know that they are now appear to be extirpated from 2 of the sites on the island where they used to occur, and the 3rd site where they were once widespread, we actually don't know anything about. But are the blue-spotted salamanders on Pelee Island eligible for a status assessment? Well, I think we can meet this criteria of taxonomic distinctness. They have to be discrete and significant, and we have evidence that they are genetically distinct, and this part of their population is actually naturally disjunct from the rest of their range. They are in fact significant. They're the most southerly population of blue-spotted salamanders, and they're isolated, which has likely given them local adaptations, and they fill a critical role in terms of the unisexual complex on Pelee Island. They're native, and they occur here, so yes, it does look like they're eligible. So let's dig into this point. The unisexual salamanders on Pelee Island have been identified as endangered both by Kasiwik and Casero. They've also been added to the species at risk list in Ontario. As you can see, they take a variety of genotypic forms, up to 10 different genotypes. If you lose blue-spotted salamanders, you get a dramatic reduction in the number of genotypes possible, down from about 10 down to about 3. So you have a dramatic reduction in the unisexual genetic diversity. You also lose the possibility of creating symmetrical tetraploids, which is the only way that you can get a reduction in ploidy to create those new diploid lineages that are responsible for creating new genetic diversity even within those lineages. Finally, because you have the ratcheting up in a single direction, you actually have the unisexual population becoming more similar in their ecological niche to the smallmouth salamanders, which will ultimately increase competition with another endangered species, the blue-spotted salamander. Now let's look at the status assessment criteria. Criteria A, a decline in total number of mature individuals. Yes, that appears to be the case. Small distribution range and a decline or fluctuation. Also yes. Criteria C, smaller declining number of mature individuals. Does appear to be the case as well. Small or restricted total population. Yes, even on Pelee Island, they appear to be restricted only to that northwest corner. A quantitative analysis, not yet. But the Kasiwik threat calculator, a quick look at that formula, does appear to indicate a very high overall threat impact, and there's no chance of a rescue effect. Let's summarize all of that. Pelee Island is the only one of the Lake Erie Islands with blue-spotted salamanders. The population on Pelee Island is genetically distinct from the mainland Ontario populations and likely has unique adaptations to warmer climate. There's an apparent loss of blue-spotted salamanders at 2 locations on Pelee Island over the last 30 years. Overall, there's a very low abundance of blue-spotted salamanders at all sites sampled between 2015 and 2018. We've only captured 3 adults to date. Currently, the status at the only site where blue-spotted salamanders were fairly abundant 30 years ago is unknown, and that site is unprotected. And finally, blue-spotted salamanders are critical to maintaining and generating genetic diversity in the endangered unisexuals that co-occur on Pelee Island. Thank you.
Our next presenter is Megan, and they'll be talking about whether constructed ponds meet the needs for ambystoma larvae. And so we can see your screen, you just need to start the presentation.
Alrighty, is that okay?
Yep, perfect, go ahead.
Great, so my name's Megan. I'm going to be presenting about the research I did while I was completing my undergrad thesis at Trent. I worked in collaboration with Tom, our previous presenter. And it's entitled, Do Existing Constructed Ponds on Pelee Island, Ontario Match the Habitat Requirements of the Endangered Ambystoma Larvae? So Pelee Island, as we just learned, is Canada's most southerly island, and it's bordered by Lake Erie. In the 1880s or so, over 50% of the wetland on Pelee Island was drained and converted into agricultural fields. Now, this has a variety of negative ecological consequences, but it also really impacts the unisexual or the Embystoma species that we see that live on the island, of which there are 3: the smallmouth salamander, the blue-spotted salamander, and the unisexual salamander. So the smallmouth salamander is listed in Canada, like what we just saw. And I won't get into too much detail about the unisexual salamander, but just know they rely on other species to reproduce. So it is logical to assume that a reduction in the smallmouth population would lead to a reduction in the unisexual population. However, it's not all bad news on Pelee. So once people started learning about this continuous decline in wetlands, a few stakeholders started to create what we called constructed ponds on the island. And they were created particularly for the Embystoma species to increase their breeding habitat. But as you can see from these pictures, constructed ponds and an image of a natural pond vary quite a bit in environmental characteristics. And so that's really where my project came in. We were interested in seeing if the constructed ponds replicated or provided breeding habitat for Embystoma species as we saw them in natural ponds. So we started off running some models and looking at larvae presence, and this is binary. They were either present or not present. And there were 3 environmental characteristics that predicted the probability of presence: crayfish burrow presence, canopy cover, and leaf litter. So as all 3 of these variables increased, so too did the probability of presence. Crayfish burrow presence may just be that the chimney crayfish on Pelee enjoy the same habitat as the Embystoma larvae. However, hence their name, they create these little chimney-like structures, and during our sampling, we saw that adult Embystoma salamanders often used these structures as refuge when they were migrating to and from the pond. So while it may just be that they enjoy the same habitat, it could also be that the adult salamanders choose specific ponds that have these structures available for refuge during their migration. We then ran some abundance models and found 2 key environmental characteristics that predicted abundance: leaf litter in the substrate and submergent vegetation. So as leaf litter increased and when submergent vegetation was present, we also saw an increase in abundance of the larvae in these ponds. So now that we knew what environmental characteristics were most important to predicting presence and occupancy, or occupancy and abundance, we wanted to know if the natural ponds and the constructed ponds really differed, and if the constructed ponds actually did provide suitable habitat. So we ran a PCA analysis, used the PCs that explained more than 10% of the variation in the data set. Following a significant MANOVA, we ran an ANOVA and found that PC1 best determined the variables that differed between pond types, so constructed and natural ponds. And as you guys will see on the slide there, PC1 had all the variables that represented those that determined occupancy and abundance. So high canopy cover, leaf litter, presence of submergent vegetation, and interestingly, cooler temperatures. So at the end of the day, what does all of this mean? Unfortunately, it means that the constructed ponds we see on the island right now aren't replicating the natural habitat and the required habitat for Embystoma breeding. However, that doesn't mean they never will. There were quite a few studies I read that showed anywhere between 5 to 10 years, and sometimes upwards of 20 years, was enough time for a constructed pond to naturalize and start to represent the habitat that was actually required. Also, it means that when we are creating constructed ponds, wherever we are, if they are being constructed for a target species, you have to take into consideration the ecology and the life history of that species. Otherwise, you may end up creating ponds that aren't successful and are never colonized. So with that, I want to thank everyone for listening, but also thank you to the specific people listed on this slide because they definitely helped my thesis come underway. As well, I'm happy to answer any questions in the session after this.
Fantastic. You got in under time. All right. So our next presenter will be Ryan to give us a talk about blue racer habitat management on Pelee Island.
Thank you. I'm going to share just not share my notes or even try to, so I don't have any problems. So hopefully I don't ramble and go over time, but I'll try my best. Okay, so hi everyone. My name is Ryan Wolf. I'm a master's student at the University of Toronto in the Rawlinson Lab. And today I have the privilege of wrapping up the CHS conference talking to you about my research projects on Pelee Island with the blue racer. So for those of you who don't know, the blue racer is a subspecies of the North American racer and is listed as both provincially and federally endangered. Now, they only ever existed in Canada in this extreme southwestern tip of Ontario, and in 1983 they were extirpated from the mainland and now can only be found here on Pelee Island. So all the studies that we have on racers so far occurred from 1970s until about 2002. And this told us everything we know about them currently ecologically, as well as their range of occurrence and the numbers that still existed on the island. And since 2002, no real research has occurred, and that all kind of stopped. So that brings me to my first project, where the purpose is to produce a new population estimate to know how many individuals we have left on Pelee. In 1995, Ben Porchuk produced a population estimate of about 250 adults left and noted that this was rapidly declining. Then Rob Wilson did another study in 2002 where he said there's about 140 adults left, but this was likely a bit of an underestimate since they weren't able to access a couple of key areas in their surveys. But then after 2002, we don't really know what the population status is for this endangered snake population. But we do have an identified significant decrease in the number of observations during yearly opportunistic surveys conducted by Jeff Hathaway and crew that he takes down to Pelee every May 24 long weekend. So I'm trying to end this almost 20-year knowledge gap by doing intensive daily surveys with my team across Pelee Island for the 3-year period of 2020 to 2022, where we capture racers, measure them, weigh them, and pit tag them, so we can identify any recaptures over the rest of the study and produce a new, much needed population estimate. And the second project that I'm focusing on is assessing habitat management strategies for the blue racer. So the species relies on open canopy grasslands such as savannas and alvars, and most of this habitat type has been lost over Pelee Island's natural areas in recent decades because they've transitioned into the dense shrub thickets you can kind of see from the examples in the pictures on top there. But thankfully we have a few conservation initiatives taking place where prescribed burn and mechanical removal of woody vegetation are occurring to try and restore some of these grasslands for the species. So my aim is to compare the efficacy of each of these management strategies for the blue racer using 3 different metrics. The first one is thermal quality, where I have copper snake models placed out in a series of control and treatment sites on Pelee Island, and they all record the environmental temperatures that the snakes would experience if they were to be in that microhabitat in which the thermal model is placed. And so the data that I'm collecting from these models should be able to tell me any changes in the thermal quality of the habitats associated with prescribed burn and mechanical removal treatments. The second metric is doing habitat suitability, where I'm determining changes in the vegetative structure and the associated habitat availability for the blue racers with each of those management practices. And lastly, I'm monitoring the snakes themselves to document any relative abundance changes after those areas have been restored to see if they're using those lands more than they are the unaltered ones. So I'm still in the middle of my work. I have about 2 years of data collection left for some stuff. And so I decided not to really show any of my results yet, but hopefully I will at the next CHS conference, and hopefully that can be in person too. But I do want to highlight 2 things. So one is that by the year 2025, it was predicted that blue racer could potentially be extirpated from Canada based off of the initial surveys done in 95 and 2002. But I'm very hopeful and very confident based off of capture numbers from this year and last year that they will not be extirpated by 2025 and that they're actually doing better than we thought they would be at this point in time. And the other thing is, by the end of the work, I want to release habitat management guidelines for conservation landowners for blue racer, which will hopefully increase our conservation capabilities for the species in Ontario and Canada. So I want to say thank you, of course, to Niall Rawlinson and the University of Toronto for their support. And I want to give a special thank you to Tanya Pulfer and the Ontario Nature staff, to Jeff Hathaway and the Scales Nature Park crew, Jen McCarter at NRSI, and that lovely group of individuals in the middle picture there for all of their work on Peely Island this summer. Amidst a global pandemic, you guys were freaking awesome and really became family. So thank you so much.
All right, thank you, Ryan. So yes, it's been a lovely active chat so far, but if you have more questions, please add them in. We'll start with a question and a bit of a comment for Nick. You got lots of congratulations for describing a very complicated topic. And then
I'm sorry for cursing.
That's a bit, we all forgive you. And so what do your findings mean for species divisions?
So that's some of the stuff that feeds into some of the contact zone work that I've done. We'll probably try to elevate the Western lineage to a specific level because it seems reproductively isolated, not just by call, but the contact zones are very narrow, like equivalent to Bombina fire-bellied toad widths. So it seems like it's fairly strong reproductive isolation between that Western lineage and the rest of them. But the rest of them just seem to be a big giant smear of integration. Peepers don't seem to mind. So just the ones in the hills and the interior highlands.
Super interesting, especially for those of us with less knowledge than you especially. Next, let's go to another comment for you, Tom, that Jackie shared an opinion I think a lot of us share, that is kleptogenesis is such an interesting reproductive strategy. And then from Pam, how did Peely Island form? She's curious about its age, if it's 4,000 years old.
Yeah, I'm no geologist, but what I understand is that essentially these areas were elevated. And as the glacier started to melt, they essentially backfilled all of those areas in and around where the islands are today. And so basically the rising water levels eventually filled in what is now Lake Erie, and those are the high points that are left over after that happened. So yeah, so the best estimates that I've come across are around 4,000 years, but some people say as long as 6,000 years of isolation there. But Peely Island's interesting just because it's so big. It's also the furthest from the mainland of all the big islands. So there's some other big islands like Kelly's and the Bass Islands, and they have a lot of ambystoma that we don't get on Peely, but they are much closer to the island. And that paper, the King paper that I showed at the beginning, has a really cool island biogeography explanation for all of that. But it basically conforms exactly to the predictions of island biogeography, which is pretty cool.
Okay, next for Megan, we have a question from James. Do you think it is pond construction per se, or more about where people construct ponds, for example, further from closed canopies, which means warmer temperatures, less leaf litter, etc.?
Yeah, so I think it's a little bit of both. If we were able to construct ponds close to already existing and pre-colonized ponds, that's a great way to increase colonization. And then you have the suite of environmental factors that you already know are important. So for example, if you have a colonized pond, ambystoma salamanders and amphibians in general have really high site fidelity. So if they're born in Pond A, they're going to try to return to Pond A for breeding. So that in and of itself is a really big struggle when you're creating constructed ponds specifically for amphibians. And these ambystoma salamanders have relatively small dispersal limits, even for breeding and migration. So definitely where you create the pond is really important. I think one of the issues, not necessarily an issue, but it's definitely a struggle on Peely, the Nature Conservancy of Canada is a really big stakeholder that's created a bunch of the ponds. And they're able to buy up abandoned agricultural farms that used to be pre-existing wetlands, but because the land they're buying is an agricultural land, it's flat and like there's not a lot of forest. It will become good wetland in the future, hopefully, but it's kind of an issue of figuring out how much restoration and reclamation work and money you can actually put into that versus just hoping that it'll naturalize over time. But again, when you're working with an endangered species, that like 20-year naturalization period isn't always realistic.
All right, thank you. From For Ryan next, we have a question from Anne. Have you found any hibernacula for racers, and what do they look like?
Yeah, so there's two questions. I want to answer John's first. John Jacques, is that okay? So I'm not sure I'm actually allowed to say how many captures I have exactly. It's funny because...
Sorry, what is the question for everyone?
Oh, sorry. So John asked how many racer captures and individuals in 2020. And so it's funny because Nick actually asked me this the other day, and I'm not sure I'm allowed to disclose that information exactly, but I can say that in 2020, the number of live captures that we had was tied for the most number of live captures that they've ever had on PLE, including the Ben Portek study in 93 to 95. So pretty good. The age distribution is a little bit different though. To answer Anne's question about have I found hibernacula? Yes, we have many identified hibernacula on the island, and they range from, usually they're just fissures and cracks in the limestone bedrock, but sometimes they use foundations that are old and abandoned. And I don't think any of them have been documented using like mammal burrows, but they also use the old systems of tree roots to access the cavities that are underneath the bedrock, underneath the Alvar limestone.
Oh, interesting. So Anne also put in some more comments in the chat. And thank you for doing my job better than I can. I'm sorry, John, I missed the question. But I think the question that Pamela had was sort of answered in the chat, but has the human population on Peely Island increased? And the answer was decreased, but cottagers and day visitors have increased, and people are nodding who've done the work there, so I think we're good. And then we got a question for Megan from Patrick, and it's a bit of a mix of a comment and a question, I think, but crayfish burrows as substitutes for the more typical small mammal burrows as terrestrial refugia for ambystoma, sorry, that'd be neat. And then Steve was also saying that crayfish are used by lots of different animals. So Patrick, maybe if you jump in, was it a question?
I suppose it was amusing, amusing in both senses. Amusing, really just the sense that it seems like a very interesting. I've been doing lots of work reading about small mammal burrow use by ambystoma and how that relates to microhabitat that they experience. And I just thought that it's a neat analog there. So thanks very much for sharing, Megan.
Yeah, no worries.
All right, and you have another question from Amanda and Matt. The last photo that you showed of Laterale, was it taken from Peely Island? Because the moss, oh my goodness, why? It looks like it might be Brio Andersonia illicebra, which is endangered. I hope I got that.
It was from Peely. I don't know if it was actually a Laterale that we took, because most of the species we can only differentiate based on genetic analysis, but in terms of the picture, yeah. It was on Peely. I can send it to you if you want to send me your email, but I don't know anything about moss, so I can't answer that question.
Is that spoonleaf moss? Is that what the concern was?
Yeah, spoonleaf moss.
Yeah, I thought so.
Well, good luck. And another one for you, Megan. Are there any concerns about pesticides on the reclaimed land?
So that's not something that we looked at specifically, so I can't say yes or no. I would assume that it is something the NCC is taking into consideration when they bought this land, but I don't know if those agricultural lands used pesticides or what they used. Tom might know, but it's not something that I looked at specifically.
Yeah, there's lots of room for investigation there. We don't really know the answer, how much contamination there potentially is existing or not. I certainly don't have the answer, but it'd be exciting to find out and interesting.
All right, and Jordan is wondering how the hydrology of artificial hibernacula on Peely compared to natural hibernacula used by racers in the States. If you have any idea, Ryan.
No. It's funny. It's because a lot of time people don't actually study the racers in the States, even though they're can arguably be considered one of Canada's most endangered snakes. They are super common in the States, and so not a lot of research actually occurs on this species, or this subspecies, I should say. So I don't think they have information on that, and I haven't monitored the artificial hibernacula that are on Peely in terms of hydrology. All I know is that they flood out and they're completely covered by water in the spring. But Anne, I don't know if Anne Yegi, I think she was doing monitoring of that on Peely, potentially.
Well, we just built 3 new ones, hopefully communal type that we're testing. They're not open for snakes, but they were built near the existing ones that were built at the Florian Diamante site. So those sites, unfortunately, only one was actually constructed. I'm sorry, I'm going to let NRSI spill the beans for them, but only one was actually constructed with a cavity. The one that was flooded, that had the dead rabbit floating, you know that story. The other 2, they never built a cavity. They didn't put any rock cavities in there. So there was no space. It was just pipes into dirt because we excavated them. So that's news, and that's hot news. And anyway, we'll let you know. I'm working with Jen McCarter on that, and we'll let you know how they work out. But right now we're just monitoring temperature and water levels in the newly constructed ones that we just built this fall. So we'll see what happens.
All right, that wraps our 3rd session. Thank you, everyone.